Right. Good day, everybody. Um, this is basically officially my first live on YouTube. I'm working with StreamYard because I want to share the screen as well. I want to discuss some materials whilst uh, doing this kind of vlogging. Um I aim to do this kind of a scientific vlog. Maybe I'll change the name of the channel as well to a more relevant one. I will be concentrating myself upon immunology, biology, and pathology, and I also have a particular interest in molecular biology. For those who don't know, I'm a master's student in biomedical sciences, and I have a particular project in COVID 19. Specifically, I'm interested in the therapies that help mitigate the disease. Uh, and obviously, we got to look at the vulnerable groups. We got to look at the context, and we got to look at uh, where this virus came from, how it adapted to the environment, and how it is causing disease. And it causes disease using very important components of the innate immune system, which is the cornerstone of the immune system. So let's get going. But first, I want to check whether I have a sound because yesterday I tried to go on a live stream and I couldn't have a sound. Like I couldn't hear myself. So just bear with me for a sec. So yes, I'm live right now. Perfect. Right. So we're going to begin with the definition of the immune system. The immune system basically is there to protect you from disease, from pathogens like viruses, bacteria, fungi, protozoa, and so on and so forth. And you encounter a lot of pathogens every single day and even every single hour. So the immune system is there to adapt itself to new pathogens coming in and training cells, immune cells, to respond and build antibodies, basically. So you see here in the right hand side, you have the adaptive immune system. They also call it a specific immune system, which consists of T lymphocytes and B lymphocytes. B lymphocytes, what they do, they adapt, they become plasma cells before they produce antibodies. And developed B lymphocytes are of two types. You have the plasma cells and you have the memory cells. The memory cells do not produce antibodies, but they're there to basically produce memory, to remind itself of the pathogenic history. And T lymphocytes, I want to just come to the two most important T lymphocytes that we have is the CD4 plus T helper cells and the CD8 plus T cells, the cytotoxic cells. Um, and an immune response, a full and healthy immune response, starts from interferons and other chemicals that are produced by cells to send signals basically to the first and second line immunity cells, which are some of them are natural killer cells, other of them are macrophages. Then you got the mast cells, and you also have another one, dendritic cells. Um, macrophages, and other reticulum cells. But the most important ones are these four. The natural killer cells, what do? As soon as they receive a signal from type 1 interferons uh, from the infected cells, they travel to the infected cells and neutralize it via induced apoptosis. Um, and then you've got signals going to macrophages and dendritic cells eventually, which are antigen presenting cells. What do these cells do? They present antigens to developing T cells, and then they will differentiate. Some of them will become CD4 plus T cells, which will help with antibody responses, and others will differentiate into CD8 plus cytotoxic T cells, which they will travel to the infected cell and have a similar activity as the natural killer cells, which is to induce apoptosis of the infected cells through a series of enzymatic actions. Um, so chemicals like type 1 interferons, they are the core of the immune system, not because they're part of a non-specific immune system, but because there is a link that is critical between adaptive immune system and the innate immune system. Without that link, you don't have a good adaptive immune system response at all. So let's get started really. Let's go to figures. I want to show you some figures to explain type 1 interferons, how important they are. So I will be starting with the intracellular production of type 1 interferons. It all starts from these receptors here. They're called pattern recognition receptors. And what do? As soon as the virus enters the cell through the ACE2 receptor, they normally start producing signals which are sent all the way to the nucleus. And I want to emphasize on IRF3 and IRF7. And just zooming the screen for a little bit so everybody sees this properly. So signals are sent all the way from the surface of the cell, from the endosome, and also from the mitochondrium. Phosphorylated signals are sent to the nucleus. And then these signals, they act as transcription factors, which activate genes specifying type 1 interferons. And then these type 1 interferons, what do these genes do? They synthesize the protein, because type 1 interferon is a protein, but they synthesize it through mRNA. So you have interferon alpha and interferon beta, which are type 1 interferons. You have the transcription process, and then you have the translation process, which is inside the cell. It's not outside, but this is like a brief description of the process. So once the proteins are produced, they undergo exocytosis, they exit the cells, and then they travel to a cell specific for building signals against viruses, uh, restricting inflammation. And as you can see here, in this context, you have signaling against cancer. So type 1 interferons, they're not only very important for treating and preventing infection, but they are also important to lower the incidences of cancer, to even prevent people from developing cancer. Through what processes? Basically, there's a series of processes, but basically a normal signaling of type 1 interferon will induce apoptosis of cells that have mutated DNA. And that itself um, will restrict the growth of cancer and will often even prevent cases of cancer like malignant tumors, which can be disastrous often, depending on the type of cancer. But malignant tumors are quite dangerous, to be honest. Right. So let's go to the signaling. So this is another diagram of intercellular production of type 1 interferons. So here you have, they call it interferon 1 pathways. So once the interferon 1 molecules have been uh, have exited the cell through exocytosis, they bind to a receptor, which is the interferon alpha beta R1 R2. So it's a dimerized receptor. It's a dimer. So outside you have these structures that bind the protein, and inside you have the JAK1 tyrosine kinase 2 dimer. And from there you have four main pathways involved in different kinds of immune responses. But in this material, I will be focusing on the two on the right hand side, which are the antiviral responses and the anti-inflammatory responses. These two pathways alone can play a critical role in mitigating the effects of the current pandemic. And as a master student in biomedical sciences and somebody who is personally passionate about immunology and about pathology, I don't understand why there is only a little talk on the public arena about how type 1 interferons can change the game really of this current pandemic. Uh, Next one, I'm going to basically describe the three scenarios with interferon 1. Just bear with me for a second. So you have a scenario that the first scenario includes a robust response of type 1 interferon and a great quantity, really, a large quantity of synthesized interferons which travel to a multitude of cells that facilitate signaling to important immune components from the innate immune system, like natural killer cells and macrophages, as well as dendritic cells, all the way to the adaptive immune system, including B cells, plasma cells in this case, and the cytotoxic T cells with CD4 plus helper cells, which help antibodies have a proper response against the virus. So if you have an early response with type 1 interferons, you have an extremely low chance of developing severe sickness, and over 99% of the cases, even 99.9% of the cases involving an early response with interferon 1 will result in mild to no symptoms and generally no complications. The second scenario is where type 1 interferon will be produced and will be exocytosed, but the response will be delayed and sometimes it can be exaggerated. But the problem is here that even if it's exaggerated, there is no effect. On the contrary, there's a worse outcome of the disease. So it's very important to focus on early treatment and prophylaxis when it comes to the current pandemic. Some hypotheses circulating mentioned that type 1 interferons could be administered in higher dosages even later in the disease stage, but personally I'm skeptical about that one. And more specifically, in the second scenario, you have generally moderate to severe sickness. You have a number of visits in the intensive care unit. And yeah, a few cases even include some need for respiratory support. The first scenario involves a completely absent response of type 1 interferons. And as a result, there's a lot of cases of severe illness. I would say most of them are severe in multiple aspects, not just in a respiratory manner, but it's beyond that. It affects more organs. The viral load is that big, is that high, that viruses cross into the blood, causing viremia, and systemic inflammation likely will occur. You have all these secondary bacterial infections as well. You have acute lung injury, acute respiratory distress syndrome, and once the virus gets into your blood, you may have blood clotting, you may have inflammation of nearby organs, and sometimes even organs that are not that are far away from the lungs, and that may be important for your health. Um, a lot of mechanical ventilation needed, many deaths. So, given this evidence of scenarios, practical evidence, including dozens and dozens of clinical trials, which I'll be explaining soon, I have reasons to believe that we might be able to build a, a mechanism based on natural immunity in type 1 interferons or interferon alpha and beta to simulate infection and stimulate a full and intelligent immune response at the same time. Um, they've been saying that doctors and scientists, my apologies, have been discussing about creating nasal sprays that contain type 1 interferons, interferon alpha, and interferon beta. And given the evidence, given the results of the plenty of clinical trials, multiple types, and studies on human cell lines as well, I believe it is safe to say there is a likelihood of effectiveness as a vaccine, even a vaccine, because of this simulation of infection. Uh, Mainly, you could talk about prophylaxis and early treatment, um, but the fact that this compound um, trains the immune system to to become better, because that's basically part of the vaccination scheme, is to help the immune system build responses and build memory. Um, and everything starts from interferon 1, to be honest with you guys. So if you have a healthy type 1 interferon response, you automatically have a sharp adaptive immune response. You have very good antibodies, you have properly trained T cells, you have properly uh, divided and trained plasma cells and memory cells. So that itself, like, vaccination is about building an adaptive immune response. Yes, definitely. But if we start from the type 1 interference, we might be getting even a better adaptive immune response and adaptive immune memory. So, yeah, we should be taking this into consideration, I believe. Um, a low dose would be, in my opinion, more effective and safer than a high dose. A low dose involves like nasal spray, and a higher dose includes uh, involves an injection, intramuscular injection. And here's another thing: we need to be focusing on mucosal immunity, not intramuscular immunity, because the virus first infects the nasal pharynx, and as a result, mucosal immunity is involved first hand here. And mucosal immunity has uh, immunoglobulin A. Immunoglobulin A, or antibody A, is very important as well in the defense against SARS-CoV-2. So let's go here to specify a little bit about the timeline of the immunity. Here, in this case, you have an immune reaction against the HSV2 virus. So once it enters the host cell, you first have the interferon beta response. And this is the most important part, this part and this part, because these two, they stimulate proper adaptive immune responses. They regulate inflammation and they build intelligent responses against the higher viral loads. They restrict the replication and spread of the virus quite significantly, to be honest. And yeah, you have inflammatory monocyte recruitment, natural killer cell recruitment. They have interferon gamma, which is also quite important. It's a type 2 interferon, but that one is not restricted by the virus. And then you have the antigen presenting cells, which are, like I said, macrophages with dendritic cells. Major histocompatibility complex expression. So with CD4 plus T cells or T helper cells, you have the major histocompatibility complex type 2. And for cytotoxic CD8 plus T cells, you have a major histocompatibility complex type 1 expression with antigen presenting co-stimulatory molecules that they are part of the mechanism to present antigens and train the T cells to differentiate and respond. Now I'm going to talk about how just let me stop sharing the screen for a while. So now I'm going to talk about how SARS-CoV-2 and influenza A viruses, how they how do you call it? How they restrict type 1 interferon responses, how they impair these kind of responses often, and how they cause severe disease for that pathway. So because SARS-CoV-2 and influenza A produce some sort of accessory proteins, for example, non-structural protein 1, and in the case of SARS-CoV-2, you also have the non-structural protein 16. Because of these kind of proteins, you have more frequent scenarios where you either have a delayed type 1 interferon response or even a completely absent type 1 interferon response. And as a result, you have severe illness and death in many cases, much more cases than needed, than desired. How can I say this? Much more cases than expected. My apologies. It's my first sort of live. So yeah, how do these proteins work? Non-structural protein 1 is a more dangerous one because it more directly impairs the response with interferon 1 than non-structural protein 16, although both play an important role in restricting these kind of responses and impairing immunity altogether. Non-structural protein 1, it binds to the RIG1 pattern recognition receptor, which is important in sending signals to genes that activate the transcription and translation of the protein. And uh, the, how do you call it? The non-structural protein 1, it also impairs um, the translation of the protein by binding to the ribosome, by cutting the messenger RNA, and by canceling the translation process of type 1 interferons. And as a result, because of these two non-structural proteins, you can have a significant reduction in the synthesis rate of type 1 interferons. And as a result, uh, immune cells will no longer recognize infected cells, quite a number of them, as infected. And so the immune responses will be much longer. Generally, the um, sorry, just give me a second. Um, so the immune system will have a much longer response. Uh, there will be a much longer time when there will be no symptoms, but actually there will be inflammatory processes already activated. The immune system will be basically confused. Uh, there will be a delayed response or an absent response, leading to severe cases, leading to hyperinflammatory reactions and to high viral loads. With influenza A, the reason why it's milder is because they don't produce that many non-structural protein 1 copies, and they also don't produce a non-structural protein 16. So that's one reason why the flu is less deadly than COVID-19. Um, we got to think about a future here because uh, they keep telling us about influenza A new strains, the avian flu causing strain, which is the influenza A variant H5N1. And this is very important, like, because you have a lot of evidence that it is type 1 interferon based therapy and prophylaxis is essential against these kinds of viral respiratory based disorders, and even against cancers, against neurological disorders, and against many types of pathogens. So building up, like creating this sort of nasal spray containing type 1 interferons could really turn the situation around, could make us more prepared for these epidemics in the future and for even pandemics in the future. Let me just share the screen again for a little bit. Just give me a sec. So I'm just wondering whether I could just select a page, but I can't. So we just have the title here, Interferon 1 nasal sprays, a good-looking COVID-19 vaccine candidate. There's two of us at the moment who are working on this research proposal. Just add an introduction here. There's new evidence about the source of the virus not being fully natural. In fact, there are materials that are mentioning a very high likelihood that it's not completely natural. Like it may be 99 or 99.5% natural, but even 0.5% could be important in science. Not to criticize the concept of immunization, because immunization consists of uh, ways to support the immune system, build memory, and so people don't get sick. But here we might have a quite different, how do you call it, context. And I believe that because the virus specifically produces, um, the virus specifically produces gaps in the innate immune system, I believe that the only way we could fully outcompete it short term and long term is by developing ways to filling these gaps created by the virus in the innate immune system. Again, not because it's non-specific, but because it's the core of the immune system. Let me just go to the references for a little bit. We have the conclusion here, and we have the bibliography. We have a number of references, which include the clinical trials using type 1 interferons, interferon alpha, interferon beta nasal sprays. One of them is interferon alpha 2B nasal sprays, and the second one would be interferon beta 1 nasal sprays. Um, so, yeah, I have here quite a number of them. In total, we have like 66 proper references and two articles basically providing evidence, but not in a very academic sort of fashion. That means these two sources contain information taken from scientific papers, but the way I presented it here is not fully in an academic fashion. Like for purposes of research, I'll be thinking about maybe replacing them with the sources of the academia, basically. What I was about to say? Yeah, I was about to say about early treatment and prophylaxis. See, I have the timeline of the disease here, SARS-CoV-2 infection. Um, the peak of the viral load is usually reached around 2 days or 2 days and a half, more like in the highest, like in maximum amount of time since the first symptoms develop. And that's quite an interesting point of view, not point of view, that's a very interesting piece of evidence because when one may think that um, the disease will have like the symptoms will be most severe when there will be the highest amount of virus inside the organism, but it's not always the case. Like in this situation, the viral load is the highest when there were only quite some mild symptoms. So that's an interesting point of evidence. So using this kind of evidence, we have to say that the window of opportunity here is not that big, it's not that small either. Um, we say it's from the first outbreak, from the first hour of outbreak or the first day of outbreak until the second or the third day when the patient developed symptoms. And so in that time frame, uh, there could be a nasal spray administered to prevent the disease and to treat the disease in the cases where it needs treatment. Um, and the way we can differentiate the we can discern the right time from the wrong time, for example, is for infected people who already have had some symptoms, and if they get a positive antibody test, they should not be receiving the nasal spray because antibodies are built generally after the viral peak has been reached. Right. Moving on. we believe that the delayed administration of the interferon can uh, not only not help but also worsen the disease. Despite a clinical trial that actually showed tended to show the opposite. Yeah, the clinical trial performed in Iran, and what happened there is they used a 12 million international unit injectable interferon 1, interferon alpha in that case, and the results were very good actually. Like the death rate was cut by over 50%, and uh, early treatment had particularly great effects, like particularly sharp uh, impact upon the death rate. So that's an important thing to add here, is because it's the fact that it doesn't always happen that an intervention with interferon 1 can worsen the disease if it's um, administered later. But at the moment, we believe it's safe to say that it's only good for prophylaxis and early treatment. And obviously, here we hypothesized a number of things. Uh, First hypothesis is that we can develop an interferon alpha nasal spray using around 300 to 350 international units uh, for people located in areas of outbreak and for people who just developed the symptoms. Um, the second hypothesis is that we can combine type like interferon alpha with interferon beta, and for example have a 150 international unit of interferon alpha with 200 or 225 international units of interferon beta, and have an even better response, even better results, because although the time frame between the synthesis of interferon beta and synthesis of interferon alpha is very small, it's less than 48 hours, we believe that this small difference in time frame could still have a different size, let's say, in the success. And our third hypothesis is that we could even combine, we could even add a lower amount of interferon gamma with these two interferons, and have an even more specific intervention of interferons with interferons. Right, so I think that's my first sort of live. Let me just stop sharing the screen. So yeah, thank you guys for watching this. I would appreciate any sort of opinion, any sort of debate. Uh, I've been doing quite some research about how the disease, not just COVID-19, but also the flu, how we couldn't eradicate these disorders. And I believe that it's because of these non-structural proteins that these viruses produce, that we simply cannot build at the moment immune memory that lasts for a lot of time, because these viruses also mutate and change their antigens. So yeah, I think we should really be looking into how we can tackle the intelligence of these viruses to self-camouflage even, um, and uh, take advantage of our core immunity. Thank you very much. You guys have a great day. Goodbye.